Good morning, good afternoon, and good evening to you all, and welcome to this webinar on the future of predictive genomics in Israel. My name is Francesco Florindi. I work at Thermo Fisher Scientific, and today I will be your moderator. Predictive genomics was front and center at the European Society of Human Genomics Congress just a week ago in Vienna. Both the Lina Peltonen Award and the ESG Award were awarded to researchers performing research in predictive genomics for their achievement on GWAS studies, polygenic risk scores, and of course, genotyping. So we have to ask, is the predictive genomics revolution finally starting? The path has never looked as good, as rosy and promising as right now. But what needs to happen to really implement predictive genomics in the clinic, in healthcare? Today, we are blessed with an international panel of experts to help us answer these questions with a specific focus on predictive genomics in Israel. We will start with David, setting the scene on what is predictive genomics. Zisis and Sowmiya will help us to understand the state of the art and two crucial components of predictive genomics, biobanking and polygenic risk scores. We will move then to understanding the specifics of the Israeli panorama with our speakers, Shai and, of course, Yehudit. Dear attendees, you are more than welcome to participate to the webinar by submitting your questions for the panel discussion that will happen at the end of this webinar. Please use the LabRoots interface to submit these questions. We will answer to as many questions as time allows. This webinar is recorded and will be made available to you soon. And without further ado, I'd like to introduce now David Hanna, our first speaker. David is Director of Business Development at Thermo Fisher Scientific, and he focuses on our global market strategy in the predictive genomics space. David, the floor is yours.
Thank you, Francesco, and I want to thank the meeting organizers for having me present here. I'm the Director of Business Development at Thermo Fisher Scientific in the Genetic Sciences Group, and what I wanted to first start off is just sort of framing the problem that predictive genomics is designed to address, which is really around healthcare spend. Ultimately, we have a problem with healthcare spend globally in all countries. With what inflates those healthcare costs are spends on common diseases and the use of medications, with all inflate healthcare costs. And this next slide puts some numbers to them. So generally speaking, healthcare costs are about 10% and increasing of the country's GDP. The amount that's spent over the next 10 years on dealing with common and chronic diseases is pretty astronomical, showing this number of $47 trillion. And the adverse drug reactions using mismatching medication to patients is a leading cause of death in many countries. And all these things are inflating healthcare costs. So the idea is to move to a model in which we're actually not caring for the sick, but it's more of a preventative care model. In other words, we're managing health rather than managing the disease. So, the question is, how do we do that, and how does predictive genomics play into that? Well, one thing we know is that genetics is a large component of a large contributor of a person's overall health. So we have environmental and social factors, we've got lifestyle and behavioral choices that patients make, and then of course a person's access to clinical care. But genetics has been shown to be around 25 to 30% of a contribution to a person's overall health. And if we can manage that component better and understand what the genetics component is telling us, then it'll go a long way towards managing health. Again, rather than managing disease. So that's the question, is how do we leverage that information to improve health outcomes? That's really what the holy grail is. How to better improve health outcomes. The longer we keep our populations healthy, the better we'll be able to manage costs. So what I'd like everyone to do, and this is what I say to my children when they ask me, what type of work I do, is tell them to imagine a world in which their children might live in, which is a sample of blood is taken at an early stage in life. That is, the DNA on that sample is analyzed to assess a person's risk for disease. And we're really talking common diseases. I'll say more about that in a moment. And also a person's risk for an adverse drug reaction in which there's some known association between a gene and a drug. That information's pushed to the healthcare providers, pharmacists, clinicians, specialists, and so forth. And then that information is used to inform how a patient conducts themselves, tests they might take, tests they might not take, actually, if they have a low risk, which again, that's another thing that inflates healthcare costs, is unneeded tests. But eventually we're resulting in better health outcomes. So that's the world of the future. And it's just a matter of how quick we're going to get there, right? So what I want to say about two things that we talked about, predicting a person's risk for common disease and then adverse drug reactions. So predicting common disease risks is through something called polygenic risk scores, which you're going to hear a lot more about today. I want to contrast that with monogenic diseases. So these are sort of rare inherited disorders, some of the earliest wins from the Human Genome Project. The thing about monogenic diseases is they're generally less prevalent in the population. What we're trying to do is we're trying to tackle common diseases, those polygenic diseases. They're more common in the population. So if we can have an impact in that area, then we've gone a long way towards managing healthcare costs and just keeping our population as a whole healthier. So that's really what we're targeting here, is these complex diseases. So as I mentioned, another portion of predictive genomics, the other side of it, is predicting adverse drug reactions. So it's all about predicting, the future, really a patient's reaction in the future to disease risk and to drug responses. And it turns out that almost 3 million people, this data is from the United States, almost 3 million people have adverse drug reactions. It's 5% of all hospital administrations, and it's one of the most common causes of hospital deaths. So the outcome that we're looking for here is really a drug that is both non-toxic and beneficial. There's 3 other possibilities, but that's the one that we're looking for. And that's the ability to predict that is becoming actually it's been known for many years, and more therapeutic areas are positively impacted by the ability to make these types of predictions. A paper came out recently. We call this the Kentucky Teachers Retirement System paper, the Kentucky study, because this was an analysis of the return on the investment of employing pharmacogenomics in an actual setting. This was a 32-month trial that was done. And these bars just show that during the latter stages of the study, the average monthly medical savings was around $230 per patient. And cumulatively per patient, we see an increase in the amount of healthcare costs. In general, it's around $7,000 per patient within that 32-month study that they did. This was actually just published back in this past March, and I encourage everyone to look at it. It's a fairly landmark study that's showing that. And I'll say more about studies that are coming up in the future as we move along here. So I want to now talk about just taking a slice of where predictive genomics is happening globally. So this is a map of where Thermo Fisher is engaged globally. And some of these studies are more focused on disease risk prediction. Some are more focused on pharmacogenomics or exclusively focused on pharmacogenomics. And some are doing both and examining both, which is really the holy grail of predictive genomics. So the one that you heard about earlier, Francesco mentioned it. There was a lot of notoriety at the ESHG recently, is the FinGen study and what they're doing there. And this study, what's unique about it is that there's so many stakeholders that were brought together to make this happen. It was not only just the universities and hospitals, but actually pharmaceutical companies. And the idea here is and you'll see a theme in the next few slides, to look for reliable solutions for healthcare, develop through the association with pharmaceutical companies, be able to develop therapies that are more specialized for specific genetic conditions, which can be predicted in advance. And they used a microarray tailored specifically to the finished population. Again, the goal is just disease prevention, earlier diagnosis, and personalized treatments. It's a foundational study, and it's been going on for several years, and they're planned to renew it soon. Another very large-scale study is one that's occurring in Taiwan, the Taiwan Precision Medicine Initiative. This is predictive genomics at its core, because this is also looking at pharmacogenomics as well as disease risk prediction. Right now, there's about 15 medical centers that are involved, and by the end of this year, they will have enrolled up to 1 million patients. So they're eventually wanting to push the data into the electronic health records for each of the hospitals, and practice the model that we've been talking about, been describing earlier. So and initially, when they first got started, it was around 200,000 participants in a few hospitals, looking at a few diseases, ones that are kind of well known, and it's kind of in a pilot phase. But eventually, they want to get up to basically the entire population of Taiwan and all hospitals and all common diseases, with the goal of being able to maintain health in the population longer. A more focused study is at the University of Pittsburgh Medical Center. Now, this isn't actually gone beyond the study stage, and now they're actually implementing pharmacogenomics within the medical system, which is in the western part of the state of Pennsylvania. This, again, is focused on pharmacogenomics. The current state, they're a little bit beyond this now. This slide's a little bit dated, where there was just one main hospital in Pittsburgh and one or a few genes in 1,000 patients. They're now scaling this up. The idea is that the key thing is that collaboration in any type of pharmacogenomic study you have is collaboration with the pharmacist. So the clinicians collaborating with the pharmacist and the patient put this information in electronic health records. Cerner is what they use at UPMC. And again, matching the right drug at the right dose at the right time for each of the patients. Another study, this one has been ongoing since 2014. It's called the Million Veterans Project in the United States. It's funded by the U.S. Department of Veteran Affairs, so it's basically people that have been in the military. This is the closest thing they have in the United States to what we call a single payer system, because it's a national hospital system that includes research. The thing that's unique in the United States about this, let's call it a cohort, is that they're very ethnically diverse. So they had to be able to assess the genetics of a diverse ethnic population here. And again, the goals are to be able to better manage patients within the VA, the Veterans Administration health system. Again, it's a national a string of hospitals. But right now, they're actually using this data for some early clinical trials, both in cardiovascular diseases, in which they're trying to look at ways in which they can put in interventions earlier for people who have high risk, according to polygenic risk scores. Another one is looking at prostate and lung cancer, being able to accelerate the use of new interventions for patients that have high risk of prostate and lung cancer. Usually the introduction of the more powerful therapies is introduced later, but if we know that a patient has a high risk earlier on, then they can accelerate the use of the more powerful therapies and therefore improve outcomes for the patient. Kaiser Permanente is another study that is new to the United States. This is a privately funded study. It's about 400,000 patients that have been recruited. Again, their project goals are polygenic risk score, pharmacogenomics, looking at actionable variants. It's really more of a preclinical study to assess eventual implementation within Kaiser's healthcare system. So they're thinking in an advanced way. This is mostly centered in California. Now, for time's sake, I'm just going to kind of pass over to the last example. And at the NHS in Scotland, they're actually implementing a clinical study right now. It's an R&D study for improved medication management. And this will eventually, if the clinical trial is successful, will result in implementation of pharmacogenomics within the NHS, specifically in Scotland. They will be using a microarray-based assay and looking at sort of the low-hanging fruit in this study, which is patients that are 65 or older, 5 medications or more, and hospitalized within the last year. The output of this is a report from a company called Coriell Life Sciences, which is used, it's developed by the pharmacists and handed to the clinicians for better management of these patients. So I don't want to go over too long on time here, but this is just a flavor of what's happening globally and what we mean by predictive genomics. And so hopefully this will cue up the next speakers and go into more detail in these areas. Thank you.
Thank you, David, for this great introduction. And now that we know what's predictive genomics, we can actually move forward to understand what is the state of the art. And for this, we have our first international speaker, Zisis Kozlakidis. Zisis is the head of the laboratory services and biobanking at the IARC, which is part of the WHO in Lyon. He's responsible for one of the largest and most varied international collections of clinical samples in the world. And he focuses on, of course, gene-environment interactions and disease-based collections. A virologist and microbiologist by training, Zisis is one of the most recognized and respected voices in biobanking in the world, former president of ISBER, and of course, he has an impressive role of collaborations across the world that you can find in the full biography on the LabRoots homepage. Zisis, the floor is yours.
Thank you very much, Francesco, and thank you to the organizers for the opportunity to have a meaningful discussion. This is the way in which we are traveling. This is certainly how investments are going to happen within the context of healthcare provision, whether it is a private healthcare provision or universal healthcare provision. I think the road is going to lead us in a very similar direction. So I'm going to talk about the state of the art in biobanking. And biobanks are facilities that have been created and have multiplied over the last 10 years to support the precision medicine vision. We need to move away from the empirical medicine where we provide one treatment for everybody and move towards better targeted treatments for specific individuals at the right time that is evidence-based. But what are biobanks? And the easiest definition that we can provide is that biobanks are facilities containing high-quality, standardized, research-ready, and auditable material. And we don't only consider the physical material, just the biological samples that they contain. Equally, the data that are linked to those samples, as well as the legal and ethical aspects, so the governance of both the physical and the digital side of the samples. Over the last 20 years, the development of biobanking can be summarized as follows. Initially, we had issues with scalability. So the focus was very, very firmly on the number of specimens. Can we actually collect within a clinical setting what we're anticipating or what we're needing as high throughput technologies and equipment were made available? And very quickly, it was understood, as in many other fields in research, that we need to move away from this situation where you put rubbish in and rubbish comes out. The quality of the samples and is of paramount importance. And then the quality of the data, because we need to start implementing or at least thinking of using big data analytic methodologies. And where we are now, we're at a stage where we're focusing on the added value of the collections. What is the value of the data that we hold? But we also have to deal with complexity. When we talk about biobanks, the number of derivative samples that we can get out of a single physical sample has multiplied. The volume of data that we can get out of the the analysis that we can perform on these samples has really. So at the same time as we're saying, okay, it's really good that we have these facilities able to feed into our high throughput infrastructure in the different hospitals, we also have to grapple with the complexity that this technological advance entails. One of the ways in which we put a lot of emphasis in managing this complexity and allowing the field to progress without actually having any fragmentation is by developing a common language, if you like, a common language of standards and guidelines that can be implemented. And you can see here on the slide a summary of the last 5 years in the development of biobanking. So IARC published the common minimum technical standards and protocols for biobanks dedicated to cancer research. So this is the common denominator, if you like, the lowest common denominator for biobanks. And then we had the best practices for provide a set of recommendations as to how biobanks can improve their operations. The ISO standard was published at the end of 2018, started being implemented as of 2019. We had the BBMRI ERIC Academy come in, the qualification in biorepository science by ISBA come in 2020. So this is the side of skilling the staff to be able to respond and handle the needs that are within this environment of standards. And leading all the way until last year and this year to the development of new courses in low and middle income settings. So this is not an exercise that we perform only for high income settings. Technology is improving the lives of every one of us and has to be inclusive of all of us. So who are standards for? We can take the narrow view in terms of standards and say it's only about the internal process and the product of that particular process. But we know that once we start implementing standards, this has a ripple effect on the whole around those processes. So in the case of predictive medicine, the standard will go all the way to the donor relating to the way in which consent was obtained, the sample was obtained, and then treated, all the way through to the way the sample was released for the research user. And if we consider what the standards are addressing, again, we can have this very, very narrow view about the quality relating to the process and the product. But we know that it has a ripple effect on all the processes that somehow link to the biobanking that's implementing this standard. So personally, I cannot emphasize enough how important the quality of the biological samples and the quality of the data is in terms of the implementation within the predictive genomics field, but also the situation that they will impact all areas that relate to this type of activity. One last slide to say that this is not so predictive genomics and biobanking and the need to move forward with a more effective healthcare provision is attracting global attention. And last year we did present some initial ideas at the UN General Assembly Science Summit, and we hope to do the same this year with updates as to the progress achieved. I would like to thank you and pass on to the next speaker. Thank you very much.
Thank you, Zisis, for your overview. It's always inspiring to see the high quality work that you've been doing at international level. And we keep the standard pretty high with the next speaker, Sowmiya Moorthie. She's a senior policy analyst of the PHG Foundation. Her interest is in effective use of scientific knowledge to improve the health of individuals and populations as well. She has extensive experience in policy development for science and public health, and has contributed to a wide range of PHG projects, including work on personalized medicine, whole genome sequencing, congenital disorders, newborn screening, prenatal diagnosis, and of course, my personal favorite, work on polygenic risk scores that you can find among the resources attached to this webinar in the Laverut's homepage. So go there and download it. Sowmiya has a PhD from Cambridge and a master's in epidemiology from the London School of Hygiene and Tropical Medicine. Sowmiya, take it away.
Thank you very much, Francesco, and thank you to the organisers for the opportunity to present here. So I was asked to talk about an international perspective on polygenic scores. I think I'll give you a little bit, but maybe others can give you a bit more of a scientific background. But I'll talk to you, maybe like Francesco said, a lot of our work is thinking about the nitty-gritty implementation issues. So I will hopefully take you through some of those. Before I start, I just wanted to give you a little bit of background about PHG Foundation to set in context some of the work that we're talking about. So PHG Foundation is a health policy think tank. We have a focus on genomics and related technologies and how they can be used for better, more personalized healthcare. We've been active since 1997, so this year is our 25th anniversary year. And we carry out a variety of work funded by philanthropy, grants, and commissioned work as well. The way we function, our aim is really to help policymakers understand how new technologies could improve healthcare and the actions needed to put innovation into practice. We do this because by having a multidisciplinary team, which allows us to carry out expert assessment of the scientific basis, clinical relevance, and regulatory and social implications of novel technologies. We also work together with a variety of stakeholders, including academics, clinicians, decision makers within health systems, to try and explore some of the issues and build consensus to create robust health policy solutions. And we've been looking ahead to the future of bioscience in a variety of areas related to personalized medicine to identify technologies that can enable more effective personalized prediction, prevention, and treatment of diseases. And sort of this is the kind of framework we've been applying to a lot of our work and our thinking around polygenic scores as part of this pathway to predictive genomics in healthcare. So over the years, we've done quite a lot of work on risk prediction and its implications for healthcare. And more recently, since 2019, we've done quite a lot of deep dive into polygenic scores and their implications for healthcare pathways. And as Francesco said, a lot of our reports are available freely on our website, so please do go and have a look. Before sort of coming here today, I did a quick snapshot of research that's going on in the space of polygenic scores. And as David alluded, there's a lot of interest in using polygenic scores to enable much better predictive and prevention of common diseases. And given that all the research that's been sort of going on, especially over the past 5 years, there's been quite a bit of a step change in research in this area. There are quite a few trials. I haven't listed them all here. This is by no means comprehensive, but there is quite a lot going on in this space. There's a lot of trials within breast cancer, both in the field of screening, for population screening, as well as improving risk assessment in familial cancer clinics. There's a lot of interest in using polygenic scores to improve risk prediction in the context of cardiovascular disease, as well as other conditions such as melanoma, colorectal cancer, prostate cancer. And there are a variety of initiatives, such as those mentioned by David, and also some listed here, like Our Future Health, which are really looking to understand and unpick how we can best use this novel biomarker as part of healthcare pathways to improve prevention and care for populations and individuals. So a key question that we've been trying to unpack and understand is really whether it's clinically useful to incorporate common susceptibility variant information into prevention programs for common diseases. The value and insight that we get from rare variants, as David said, is well recognized. This is really thinking about common variants and how to incorporate that in. And some of the questions that can be broken down into are really thinking through how accurately can these genetic markers predict common diseases? Does their incorporation into risk prediction tools add value? Does using this information lead to improvements in current prevention programs and pathways? And if this is better, what are some of the organizational, ethical, and social implications that you need to have in place? So these are some of the questions we've been trying to unpack and understand. And our approach has been really to treat polygenic scores similar to other biomarkers and apply existing frameworks and methodologies to evaluate polygenic score analysis like other medical tests. This then allows us to understand the state of the science and potential implications for specific healthcare pathways, and then move towards more responsible and effective implementation. And as I said, this is really building on a lot of work we've done historically, looking at evaluation of genetic tests in the context of rare diseases. So we've been applying the same principles to polygenic score analysis. And for those of you, some of you may be familiar with this diagram on to my left here, which shows the ACE framework, which really covers analytical validity, clinical validity, and clinical utility, as well as ethical, legal, and social implications. So this is the kind of approach we've been adapting and seeing how it fits in with analysis of polygenic scores so that we can better understand. Oh, someone says they can't hear. Stop it. Oh, okay, right. So to better understand some of the implications for implementation. So, and as many people will know, for evaluation purposes and for help to sort of think through some of the evidence requirements, we really need to place the disorder and setting front and center when we're thinking about any application based on polygenic scores. And it's really important to think about linking the disorder setting and purpose, because it really allows you to then think through some of the nitty-gritty implementation issues and unpack these and sort of put in place some sort of the best way to implement these. So for genetic tests broadly, we tend to differentiate between an assay and a test. We describe the assay as a method for determining the presence or quantity of a biomarker, and the test as the use of the assay within a specific disease context in a particular population for a particular purpose. An evaluation of an assay is very different to evaluation of a test. And the reason we make this distinction is, as many people, and maybe Shai will tell you later on, polygenic scores are a technology and you can use them in a variety of contexts. So they can provide, in some senses, sort of lead to a variety of tests. And each of these will have different benefits and harms. And so to really understand and evaluate the evidence base, we need to understand the test on offer. The other sort of angle to that is that polygenic scores are computed. There are a number of steps that require to be in place in order to allow you to get that information. You need to have a process for gathering genotype data. This could be either through using a microchip, microarrays, or either through next generation sequencing. As others will tell you, there are a variety of models available across a variety of diseases. And so for health systems, they need to really have a way of identifying the best models to sort of include in this pipeline. And at the moment, models can be constructed in different ways. They've been validated to different degrees. And decision makers, our experience has been they're struggling with selecting which model they should select to take forward. Furthermore, it's complicated by the fact that models may be standalone or integrated with other risk prediction tools to give much more what we call integrated risk prediction, taking into consideration other risk factors. And then there are issues around how do you communicate this information to healthcare professionals as well as patients for decision making. So by understanding how analysis might work in practice, what's fed back, we can really unpick what needs to be in place to make sure we move towards implementation. And it's also really important for evaluation. They're quite interlinked, because to demonstrate utility, which is really think about what value this kind of information adds to a particular pathway, we really need to understand how it might be regulated in addition to how it might be evaluated. And one of the bottlenecks for novel assays and tests such as polygenic scores is that it's been quite a challenge to identify the nature, quality, and quantity of evidence required to ensure safety and effectiveness to place a test on the market, and also to place it within a particular healthcare pathway. And also, as you may or may not realize, the regulatory environment around such devices, i.e. risk prediction tools, is quite dynamic at the moment. And if we do want to move towards this predictive and personalized model of care, we need to have a good understanding of how we can get through sort of this dynamic environment. So some of the key conclusions that we've sort of come to are that polygenic scores can be a useful, some of the key conclusions that we've come to are that polygenic scores can be a useful biomarker and have potential utility. We're very enthusiastic about them, but we feel that we really need to, we're at the point now that we need to understand this test on offer and its intended purpose to make a judgment on utility and really go towards developing the evidence base for each potential application within a specified care pathway. And if we need a better understanding of these parameters, such as analytical validity, clinical validity, and utility, and what it's possible to get through existing research and what that's contributing to our knowledge base, so that we can work with policymakers and other decision makers, as well as patients, to make them understand some of the known knowns and uncertainties around use of this novel technology. And I think what we have said consistently is that there's quite a lot of emphasis on looking at polygenic score models. This is not sufficient reason for implementation. We are now, and I think the stage is shifting. A lot of people are moving towards thinking about implementation and how the test will look and how we can think about how these function in real life settings. I'm just going to end quickly by saying thanks to our team, which is multidisciplinary, as well as a lot of people that we consult in order to inform our recommendations and ideas in this sphere. And just to say that if anyone's interested in speaking to us about this and sort of having some discussions about it, we're more than open to hearing your views. And thank you very much.
Thank you, Sowmiya, for your wonderful analysis. Very, very clear, very precise. It is now time to move straight into the main focus of our webinar today, which is Israel, and to understand how predictive genomics can be implemented in the country. I welcome Yehudit Cohen back here. Yehudit is the scientific director for MIDGAM, the Israel National Biobank for Research. She has a bachelor degree in biology with a master in clinical biochemistry, a PhD in molecular genomics, capped it all by a postdoc in research in cancer. So a great background, which she paired, of course, with managing the institutional tissue bank at the Sheba Medical Center before joining MIDGAM. Yehudit, the floor is yours, and thank you very much for being here.
Thank you very much, Francesco, and everyone. Well, I will talk about Israeli National Biobank for Research as a research enabler. And actually, all what was discussed prior can be done with samples that we have, samples and data, as this is mentioned, that we have in our biobank. And I'll go through briefly about how we work and how you can use our facility. Well, this is our team. We are okay, I'll go through the hierarchy later. I will just say in few words, very briefly, that the role of biobanking worldwide is extensively important. And I won't go in details, but you can see that there are so many research works that were done using biosamples, and drugs were developed for the benefit of patients. And regarding the operating model that I will go briefly, I will just say that we have a board of directors led by Professor Chihanover, which is a Nobel laureate and responsible mainly for policies and recommendations that we need to do, and steering committee. And we also have a scientific committee that helps us decide on things that uprise occasionally. And we have 5 different hospitals, medical centers that are part of MIDGAM, and the management is responsible for all aspects of day to day. It goes through operational aspects and scientific aspects, and all the computational aspects dealing with the data and stuff, regulatory aspects, which I will mention later, and of course, budget, marketing, collaborations, everything that we believe that needs to be done. So I will go through our vision briefly. Well, MIDGAM is a Hebrew word for samples, but it is also acronyms for Israel National Biobank for Research, but it's in Hebrew. Anyway, so we are in a national infrastructure. Actually, we are also a governmental infrastructure. And our vision is to promote personalized medicine for the health and welfare of Israel's residents. And we want to, as such, we want to provide an added value and growth engine for the high-tech industry, research institutions, and healthcare organizations in Israel. I will say a few words about creation of a virtuous cycle, and I will start. I will say that everything is focused about quality of healthcare, and I will start maybe with budget, the financial support. Well, budget needs to be allocated for biobanking in order to make sure that the most relevant biosamples are being banked. And one cannot use human biosamples without ethical approval. So this is going now to the third part. And once we have everything achieved, R&D is in motion. And when a study is successful, publications will enable receiving more budgets. So this is the way we're making biobanking better and research better. And that way we can support healthcare quality. Okay. Well, I will not elaborate too much about types of biobanks. I will just say that there are many kinds of biobanks, and we are a type that is disease-oriented and project-driven. We are focused mainly on cancer, but not only. And I want to say a few words about our goals. Well, by providing human biosamples, we are an advanced platform for the biomedical research to promote collaborations between medical centers, academia, and industry, nationally and internationally. So one word that is very important is collaboration and improvement. And we also accelerate proteomic, genomic, and molecular scientific discoveries in cancer and other diseases by collecting high quality and well-annotated human biosamples. We enable the usage of national resources to develop novel diagnostic tools and biomarkers, novel drugs for targeted therapy, and the use of newest research technologies for personalized medicine. While talking about personalized medicine as one of our main goals, I want to remind us two quotes. One of Hippocrates that was mentioned before by David, I think, in a way. Well, he said it's far more important to know what person the disease has than what disease the person has. And also you said something about, this is from the European Union definition of personalized medicine, that we need to provide the right treatment to the right patient at the right dose at the right time. So I think if we're doing these, we are okay. So how can biobanks in general and MIDGAM specifically can support personalized medicine? Well, Zisis and I like this slide because it has everything in it. So we are going from, when we say personalized medicine, we mean for a group of individuals who suffer from a specific condition, there are few general alternatives. But what was done up till now is more what is called empirical medicine that It means in general that one treatment fits all. For example, in cancer, chemotherapy is a first line and it's evidence-based and it's working. But there are some patients that don't benefit from this treatment. And when we try to sort them according to specific criteria, such as specific markers, and then that it's called stratified medicine. So, all the blue people will get something that is relevant according to the markers or a gene that was found, which is good, but it's not good enough. So a higher level of therapy is personalized medicine, where the offered treatment is based on a patient's unique molecular markers. And that goes for the polygenic score, risk score that was discussed before and will be discussed later on. I will give you one example. So if there was a drug that was developed for specific indication, certain indications according to molecular marker, it can be used for another patient with a different indication, but has maybe the similar molecular characteristics. So performing researches on biobank samples is a great way to give an optimal answer to the last two options, the stratified and the personalized medicine groups. We collect, process, and preserve the following biosamples. We have tissues, tumors, and normal, fresh viable, fresh frozen, and paraffin embedded. We have blood from healthy and treated donors. We have healthy tissues maybe collected from donors which go through different kinds of procedures, maybe mammoplasty or hernia or something like that. But most healthy samples we have are from our blood samples. We also have other samples and many controls. And I want to emphasize this webinar that we recently obtained a large cohort of DNA samples from various diseases, not only oncological diseases, that can be used for research by the scientific committee, both in Israel and outside. And you can talk to me later if you want to use it. Our guidelines are strict regarding the collected samples. We never collect samples at the expense of patients. We must ensure that the diagnosis and continued treatment are not impaired. This is a major and important guideline. This is also something that Zisis was talking about before, and I can say that some years ago, friends of mine, Daniel Dubois and Peter Watson, published this article that is talking about the development of the biobanking field. I will not go into details. I just wanted to emphasize that the evolution and trends in this field are, as Zisis says before, are developing. So at the early days, biobanking was all about quantity and then quality. Later, it was mostly about sustainability and the needs that were projected by the different stakeholders. And by that, biobanks became smaller and more focused. And in my opinion, I think that the next step, actually the current stage, that is that biobanks that fit to purpose. This is our website. You can go and visit us, and we have all the relevant data collected presented there. And this, we have a database software called Data Plus, where we keep all the information regarding the samples themselves and the data of the donors. And I want to say a few words about ethics and regulations, because it's really crucial part of a proper management, especially for biobanks. So we have an IRB approval for collecting, as I said, processing and preserving biosamples for every future study. We have an informed consent form, which is 1 page, that is suitable for all kinds of studies. It enables commercial use of the samples collected. It allows withdrawal at any time. And regarding return of results, we can discuss it. It's an issue that needs to be discussed maybe later. I think I have it here. I want to emphasize that samples from MIDGAM are provided only after a designated study was approved by a local IRB. And we at MIDGAM enable distribution of biosamples to every approval Israel study. That means if you are not from Israel and you want to have samples from us, you can do that, but we will help you submit your proposal to the IRB in Israel with us. It must have an Israeli approval. Well, I think that's what I want to say here. And there's a short list of publications derived from studies of leading researchers who have published and continue to publish in scientific literature with a high impact factor that have used samples from MIDGAM. In addition, we are in contact with dozens of researchers in Israel and abroad, including a large ongoing study with the NIH. And actually, I think we'll go through the bottom line that I wanted to share with you. Please remember that we are a national and approved platform that promotes research in various fields in Israel and around the world that deals with understanding mechanisms involving cancer and other diseases, development of various diagnostic tools, development of new treatments, use of modern technologies in custom medicine, and in short, we have samples and data you need for your studies. Finally, I would like to express my gratitude actually to the donors who generously gave their biosamples and data to be used in biomedical research for the benefit of future generations. We are obligated to keep their confidentiality, and we will do our best to make their donation valuable. I would also like to thank the Ministries of Health, Economics, Science and Technology, and the professional teams of the various medical centers. And if you have questions later on, but you can keep the contact details and call us whenever you need. Thank you very much.
Thank you so much, Yehudit. Very inspiring to see the great work you've done, and also to understand that the ministry is backing you up and is making the right investments to further personalized medicine in general. And wonderful for you to remind of the real protagonists here, which are the patients and citizens we're all serving. The next speaker will. You're welcome. The next speaker will join us virtually, more virtually than the rest, because unfortunately Shai could not be with us today, but he pre-recorded his presentation. Shai Carmi is the Associate Professor of the Hebrew University of Jerusalem School of Public Health. He has an undergraduate degrees in physics and computer science and a PhD in physics from Bar Ilan University, on top of a postdoc from Columbia. He researched, of course, population statistics and medical genetics. His group, the Shai Carmi Lab, focuses on developing and applying computational methods for identifying the genetic basis of disease, for genetic testing and risk prediction, polygenic risk scores, and for studying the demography of population based on genetic variation. So thanks in contumacia, Shai, for your presentation, and I ask the team to kickstart his presentation.
Hello, my name is Shai Carmi. I'm a statistical geneticist and a population geneticist at the Hebrew University. And in this talk, I will describe recent studies on the evaluation of polygenic scores in the Israeli populations. So polygenic scores should have been explained in a previous talk in this webinar, so I'll just go over it briefly. These scores are using summary statistics from genome-wide association studies for various traits and diseases in order to predict the trait or the risk of new individuals. And the way polygenic scores are constructed is using this simple equation where the polygenic score of an individual is the sum of the number of risk alleles or trait increasing alleles carried by an individual. So GI is the number of effect alleles at a SNP, which can be 0, 1, or 2. And beta hat is the estimated effect size of that allele. So this is the main idea, and then there are many methods that were developed in recent years to select the set of SNPs for inclusion in the polygenic score, either as the set of genome-wide significant SNPs or all SNPs or some other methods, and also there are methods to refine the risk estimates. But overall, this is the basic idea. So I will now talk about ways in which we measure the performance of polygenic scores, first for traits and then for diseases. So I show here a list of a few traits that are commonly studied. And for each trait, I show you here some, like the range of estimates for H squared. This is the heritability. This is, broadly speaking, the proportion of variance in the trait that is explained by genetic factors. As you can see, it's up to 80% for height, around 50% for BMI, and so on. And R squared is the way we measure the performance of the polygenic score. It's the proportion of variance in the trait that is explained by the polygenic score. As you can see, for all traits, of course, R squared must be less than the heritability. We cannot explain more than the total heritability. And typically, we explain much less. Typically, the typical numbers are around 5 to 15%, except for height. That is the best studied trait so far, where the best scores now explain up to 40% of the variance in height. For diseases, the situation is more tricky. We cannot talk about the variance. Diseases are binary. An individual can be either affected or unaffected, so it makes less sense to talk about the variance in the disease status. So instead, we're talking about the variance in the liability. So the liability is so what is the liability? It is assumed that even binary diseases, they have an underlying continuous liability. We cannot observe this liability, we cannot measure it, but the liability is a representation of the predisposition of the individual to the disease, and only individuals that have liability above a certain threshold are affected, as you can see in this diagram here. So so we measure the performance of the polygenic score for a disease as the proportion of variance in liability explained by the score, which is typically 5 to 10% for most commonly studied diseases with relatively large genome-wide association studies. Perhaps more familiar indexes are an index for binary diseases is the AUC, area under the curve, or C index for time to event. And then to measure the performance of polygenic score, many studies also look at the odds ratio when comparing individuals at the top, let's say 5% or 1% or 10% of the distribution of the polygenic score in the population, comparing to all others or the bottom few percentiles. And also another popular measure is the odds ratio, comparing individuals who have PRS at 1 standard deviation above the mean relative to the mean. So these are common ways to measure the performance of polygenic scores for diseases. And what I will show in the rest of the talk, I will describe studies, some by my group and some by others, that measured the performance of polygenic scores in the various Israeli subpopulations, particularly focusing on Ashkenazi Jews, which is the largest population. So we know that polygenic score accuracy is lower in populations that are not European. So for example, this is a distribution of R-squared values for traits relative to Europeans. We can see that performance decreases, in particular for African population. But the question is, what can we say about the populations of Israel, as I said earlier, particular Ashkenazi Jews? So first, I will describe results for traits for height in Ashkenazi Jews from a study that we published a few years ago in the context of screening of IVF embryos. In that study, we also looked at the accuracy of polygenic scores for height, first in a cohort of 100 couples from New York, and then also in a cohort of about 30 very large nuclear families from Israel. And interestingly, we see that the performance is about the same in both cohorts, where the polygenic score for height explains about a quarter of the variance in the trait, which is comparable to the variance explained in cohorts of individuals of European ancestry using the score that we used at the time. So now, moving on to diseases. One disease that is of most interest in Ashkenazi Jews is Crohn's disease, because it has a somewhat higher prevalence in that population. So this is a study from the Mount Sinai Biobank in New York, where they evaluated the accuracy of a number of polygenic scores. As you can see here, different ways to compute the scores based on different association studies, and they evaluated the score using AUC in European, Hispanic, African American, and Ashkenazi Jewish population. And as you can see here, not only that the score accuracy does not diminish in Ashkenazi Jews, AJ here, as compared to Europeans, it is actually even slightly higher. So this is very encouraging in terms of like the ability to predict risk in the Ashkenazi population. We studied schizophrenia using an Israeli cohort of about 2,500 individuals. And in a recent study, we found that the variance in liability explained by the score is about 7%, which is again comparable to the variance explained that was seen in cohorts of European ancestry. The Mount Sinai Biobank has been very useful for evaluation of the accuracy of the score in Ashkenazi Jews because of the largest Ashkenazi population in New York. And in that recent paper, Belbin et al. Evaluated polygenic score across multiple diseases in the Ashkenazi and non-Ashkenazi individuals from the Mount Sinai Biobank. And here you can see results for 3 diseases showing the prevalence of the disease versus the PRS decile. And you can see, again, comparable, very similar performance of the polygenic score between Ashkenazi and non-Ashkenazi participants. In a recent study, we did a systematic evaluation of the accuracy of polygenic scores across multiple populations, including Ashkenazi Jews, in the UK Biobank. So using principal components analysis, we were able to identify about 1,700 Ashkenazi individuals in the UK Biobank, and we evaluated the predictive accuracy of polygenic scores across over 200 traits and compared the accuracy to the accuracy seen in individuals that were born in the United Kingdom. The results for Ashkenazi Jews are here, showing that the accuracy in Ashkenazi Jews is about 86% compared to individuals with British ancestry. So this is comparable to what we see here for Italians, and is higher, even much higher than what we see in other populations, for example, individuals from India or East Asia or African individuals. So again, all of these results so far show relatively high accuracy of polygenic scores in the Ashkenazi population, almost comparable to what is seen in individuals of European ancestry. So now I would like to show a couple slides moving beyond the Ashkenazi population and also studying individuals of other ancestries in Israel. And unfortunately, there are not so many studies, so I will just show you what we have right now. So this is an unpublished study from my lab, showing results of a study of age-related macular degeneration, AMD, from Hadassah Medical Center in Jerusalem. And here, the participants came from all Israeli subpopulations. About half of them were Ashkenazi. The rest were Jewish, non-Ashkenazi Jewish or non-Jewish. And as you can see, the Polygenic score for AMD works quite well, even in this more diverse population. In the bottom fifth of the population, in terms of the risk score, we see only 35% of cases compared to 86%. Sorry, it's not the fifth of the population. In the bottom fifth of the cohort, about 35% are cases here compared to 86% in the top fifth individuals at the highest risk score in our cohort. Now, in terms of non-Jewish populations, there is really barely any data out there. I'm only aware of one study by our group, also in collaboration with a team at the Hadassah Medical Center, where we studied cholesterol levels in a Muslim village, in which there is high prevalence of familiar hypercholesterolemia. And after we adjusted for the pathogenic variant that is common in this population, we looked at the LDL cholesterol level versus the polygenic score, and we see correlation, which was statistically significant, but the correlation was very modest. It could have been because the score was relatively old, the score that we used at this study, but it could also be that the score is simply not as predictive in this population. We don't know yet, and unfortunately, right now, to the best of my knowledge, there is no additional data about the accuracy of polygenic scores in the Muslim populations of or non-Jewish population in general of Israel. So to conclude, based on multiple studies, I think we can now say with some confidence that polygenic scores that are computed based on genome-wide association studies in Europeans have a relatively high accuracy in Ashkenazi Jews, almost the same as observed in Europeans. And this is based on both anecdotal studies as well as systematic evaluations across multiple cohorts and also across 3 countries, Israel, United States, and United Kingdom. We have very little data on the accuracy of polygenic scores in other Jewish subpopulations and non-Jewish Israeli populations. And hopefully this will change in the future, although this may be progress may be slow. Moving forward, there are a couple of small projects underway that I'm involved in for studying breast cancer and coronary artery disease in hospitals in Israel. But perhaps more promising are 2 large projects that are at different stages. One is the 10K project of the Weizmann Institute that will collect or is already collecting genetic and phenotypic data. And the second project is the CEPHAS project of the Ministry of Health that in the future will collect genetic and phenotypic data on tens of thousands of Israelis, and thereby will provide more information on the accuracy of risk prediction in the non-Ashkenazi populations of Israel. Okay, thank you for listening.
Thank you again, Shai. If you can hear us, I know he has lessons today to attend, but his presentation was really excellent in providing, again, more background on Israel. I'm asking all the speakers to join me on camera for the panel. Thanks again for your outstanding insight. And I want to remind all the attendees that they can still send questions for us to answer. We got some interesting ones already from the attendees. So thanks for being active. I will kickstart with an easy one, quotation marks, very, very not so easy, I would say, rather. What really needs to happen? We understand now the situation in Israel. We understand what's the state of the art. What are the obstacles? What needs to happen to unlock the potential of predictive genomics in Israel? And Yehudit, I would like to start from you.
Right. What needs to happen? Well, I guess first of all, we need to have awareness of the possibilities that biobanks exist, and MIDGAM, especially as a national entity, is here to help and provide the samples needed for whatever research is in place. But I also think that I'm not sure it's relevant only in Israel, but we need to maybe try to lower costs of sequencing and make bureaucracy maybe easier. I think MIDGAM can help reducing time to get those approvals needed. There is a lot of issues concerning what you do with the results. I mean, you get many results that are treatable and some that are not, and how to deal with that. I was referring to that in the presentation. I said return of results, like it's nothing, but it's an issue that needs to be discussed. I think a lot of I mean, you can divide the things that need to be done into the practical things, deal with the bureaucracy, deal with the costs and stuff like that. But on the other hand, is maybe more community things, is the awareness of the possibilities.
Zisis.
Francesco, if I may, Israel is not unique in that sense, in that during the pandemic, we've seen the benefit of being able to connect the dots in moving research forward. And we need to get into the mentality of a lot of our funders and a lot of our stakeholders that this is not only something we do during an emergency situation. This is something we should pursue as a matter of routine practice.
You're absolutely correct, Zisis. And before David, I wanted to bring in Sowmiya as well, because we mentioned the getting into the mind of the policymakers, and I know very few experts in this field who managed to communicate with policymakers as well as Sowmiya did. Can you add your perspective on this? I think we lost Sowmiya. But in that case, David, please go ahead.
No, I think that showing a path and what the benefits might look like is key. I think that was just said a moment ago, and I just want to add to that is that pilot programs need to be funded in order to demonstrate utility, in order to because it's a complicated workflow. Most of the technologies exist for doing this, and the reporting structures are mostly crafted. It's really putting all the parts together and implementing a program in predictive genomics or predictive medicine, whatever we want to call it, involves so many working parts. So it needs to be, it's sort of like baby steps. Small pilot programs need to be put in place in which the entire workflow is demonstrated, and then the key stakeholders will understand what what components need more attention, more resources, and then scaled up from there. So it's really just moving from a discussion of this is theoretical to actually piloting small programs of, maybe 10,000 samples or so, and these need to be funded. And then, and this could come from a collective funding from the various stakeholders, and then scaling up from there. It's just because of the complexity of doing something that's so, groundbreaking and pioneering. That's, in my view, that how things need to get started.
Thanks, David. So now I see that Sowmiya, she's back, so I'll move now to her. We heard from David that funding pilots would be the key. We need to explain this to policymakers. What would be your opinion on this, Sowmiya?
I'm sorry, I missed the question because my computer glitched.
The panel believes that predictive genomics is the way forward, but we need to lower the costs, make bureaucracy easier, raise awareness, and speak the language of the decision makers, the funders, to make them understand the value of funding studies that can put together the existing evidence. You are the closest, you are the one who actually managed to translate this whole problem, in my opinion, the best to the policymakers so far. What would be your suggestion to further the field?
I think one of the issues that we've consistently sort of experienced in this is that there's sometimes a bit of a disconnect between researchers, policymakers, and end users of whatever application there is. There are different levels of understanding. I mean, there isn't a shared understanding of the evidence needs at different levels. There's a different they may not be content experts. They may not understand particular things such as polygenic scores. We have experience, especially with polygenic scores, the sort of conflation with rare variants, and we've had to do quite a lot of work to sort of get rid of that misconception so that people can understand it and think about it as any other biomarker, and to say to them, actually, you don't have to be as, in some senses, the evidence sort of balance that you have, risk benefits are very different. And so there is quite I think one of the key things you can do is actually there's a lot of bringing together these different stakeholders to achieve some consensus on evidence needs, because I think definitely you can sort of almost lower your threshold of evidence needs. For example, you don't need an RCT for every single application, I think, and you have to really understand how far the research can get you, and be comfortable really that it can get to a certain point, and after that you have to just try it out and have processes in place, like you say, to feed back into the loop.
Another thing I want to also add is that what we've seen is, just a larger appetite for pharmacogenomics, just because of the proof points that was just mentioned a second ago are mostly there, whereas the polygenic risk score as a, let's say, biomarker is kind of on the vanguard of being pushed in as a clinical thing. Whereas pharmacogenomics, it's more of a well-traveled area, and for groups that are wanting to get into, predictive genomics, it seems like what we've seen is that they're prioritizing frequently pharmacogenomics as a test bed for, the connection between the patient, physician, and in that case, pharmacist. So that's just a trend that we've seen at Thermo Fisher.
I totally agree with you, David. I think both, everything is about money eventually. So we need to decide, we as a society need to decide where we put the money, either in the personalized medicine, specifically individuals, or we take something that we can help more than one, or a large cohort. So this is, it's a balance between those two, and these are the stakeholders that needs to decide where you put the money in. And the other thing I wanted to say, that as a biobanker, I can say that the data that comes with the sample, not for the sample itself, this is kind of easy, but the clinical data that arrives with the sample is so important, and this has also many problems with the harmonization of data. But it's not only the data that come with the sample at the time of collection. It must follow with the sample, with the donor for, I don't know, for as long as possible. And then you can make the conclusions of any research much more thick, and would be much more important and can be useful for the benefit of other patients and not only the donors themselves. So this is also something that needs to be considered.
You make a great point, Yehudit, and right now I'd like to bring in Shai, who actually answered some of these questions earlier. And I think he makes a very interesting point on exactly on the issue of data, particularly in Israel.
So the HMOs I already mentioned, but beyond the fact that the healthcare, that everyone is covered in just one of those 4 HMOs, the HMOs also have a longitudinal electronic medical records going back 20 to 30 years, and very detailed and very rich. And these can be, these are great for research, and these records can be used to validate polygenic risk scores and implement them and follow up the patient. So this is really fantastic and could accelerate the implementation of predictive genomics. I think in the Israeli population, generally speaking, there is a willingness to get tested. There is also interest in the genetic testing, and we see that in the program for preconception genetic screening, which is very common in Israel, and there is a very high rates of uptake of this testing, and same for prenatal genetic testing. So this is probably going to be accepted by the population once it's offered by the healthcare providers. On the other hand, weaknesses of I mean, the difficulties that we are going to have towards the implementation of predictive genomics in Israel is the little data that we have on the non-Ashkenazi population, and also the current legal system, which makes it somewhat difficult to conduct genetic studies in Israel, in particular in the academia.
So we heard from Shai that indeed the healthcare record system, he mentioned the HMOs, the specific healthcare system you have in Israel, led by several insurance companies. That's a very ripe environment where you can really connect a lot of the data that you were mentioning before. What do you think about the willingness of people to actually share their information?
So I can add that we made a short questionnaire in Israel regarding this willingness to not to share the data, well, also to share the data, but the main question was, are you willing to donate samples, share data, and receive back information? So from around half of the people that were asked were willing to donate samples, and from them, 40% wanted to get back results and were willing to share whatever is needed along the way. So I think in general, once you decided as a patient or a donor or okay, I don't like the word patient in this context, but as a donor, you decided that you want to donate your samples. So almost 40% would like to share their data and would like to get back the results for their own and to get that benefit out of their donation.
Thank you very much, Yehudit. We're approaching the end of our webinar. I would like to ask one of the questions that actually we got from the audience on developing countries. So the question comes from Muhammad Moidin Uddin in Rawalpindi, and he's curious to understand what's the panel opinion about implementing precision medicine and predictive genomics in developing countries. Now, this is clearly not the case of Israel, but we know that a lot of developing countries are looking at Israel and Europe as examples for the future of their home healthcare systems. Zisis, I would like to come first to you and then to Sowmiya and the whole panel.
Yes, absolutely. I'm in agreement with what you said. I think Israel can serve as a blueprint for how things should be done, because we've had experiences of actions that have worked very well and actions that have worked less well in the past and even in the recent past. I think what we want to avoid is technology becoming yet another differentiating or discriminating factor when it comes to low-middle income settings. There are examples that we can use, say from Israel, in developing infrastructures that serve as a central center of excellence and cover a particular part of the population, but do it very well. And that can be an example that then you can use to roll out to the remaining population within that particular geographic area. So working from something small and well-defined that works very well, and then expand it as time goes on.
Thank you, Zisis. Sowmiya.
So I'd say actually there's two specific angles. One, like Zizi said, I agree, there is looking to learn about infrastructure and looking from other countries and how you set this up. Broadly speaking, a lot of developing countries have a very different healthcare system, so they have to develop something that's specific to theirs. There's also some consideration around the underlying science and the data and what it can tell you and how it can apply to these populations, because as we know, there's a lot of lack of diversity in genetic studies. So I think as of June 2021, 86% of genetic studies were in European populations. And so we really need to understand what a lot of this information means for other countries and other populations. Even studies that have been carried out in African Americans are not completely transferable to those in Africa. But, there are efforts underway to improve this, and I think they will sort of help develop such systems. But it is, I think, there's an opportunity there. I think definitely predictive genomics, personalized medicine is applicable to all countries everywhere. You just have to look at how it fits in with emerging systems and how best to integrate it in.
Thank you so much, Sowmiya. We're really, really close. At this time, I think that we don't have time to answer all the questions, but thanks again to all of those who submitted questions. Our time is up. I would like to thank you first and foremost, our speakers, for your time, your expertise, for sharing your invaluable insight into predictive genomics. I'd also like to thank the team who actually made it, put all this wonderful webinar together. As I said, this is recorded. It will be available online for you to share with your colleagues and your teams very soon. This is actually Predictive Genomics Week within Thermo Fisher, I feel. Later today, we have another webinar on the more technical aspects of delivering predictive genomics, focused on really the difference between the different technologies you can use to do predictive genomics. You can find the webinar on LabRoots. Just search for Predictive Genomics or Thermo Fisher, and you'll find it. Finally, you might also be interested in the next country focus on predictive genomics. We'll look at what Qatar is doing on the 28th of June. Again, on LabRoots, so you can use the search engine within the system to find it out. And please don't hesitate to reach out to myself, to the team, and of course to the speakers via the LabRoots contact form or via